Welcome to Public Health on Call, a new podcast from the Johns Hopkins Bloomberg School of Public Health. Our focus is the novel coronavirus. I'm Josh Sharfstein, a faculty member at Johns Hopkins and also a former secretary of Maryland's Health Department. Our goal with this podcast is to bring evidence and experts to help you understand today's news about the novel coronavirus and what it means for tomorrow. If you have questions, you can email them to publichealthquestion@jhu.edu. That's publichealthquestion@jhu.edu for future podcast episodes. Today, I speak with Dr. Emily Erbelding, the director of the Division of Microbiology and Infectious Diseases at the National Institutes of Health. Dr. Erbelding leads a huge part of the scientific effort in the United States against COVID-19. We speak about basic research, clinical research, and vaccines. Let's listen. Dr. Erbelding, thank you so much for joining me. It's been probably about a decade since we worked together in Baltimore, and I remember you as the infectious disease expert at the health department who knew the answer to every question about every infectious disease that came up. And now you're the division director for microbiology and infectious disease at the National Institutes of Health.
Yeah, thank you, Josh. It's I don't know if I knew everything, but I tried.
So you you left Baltimore and you went to the NIH, and now you're in this position where you're responsible for the work of the National Institutes of Health supporting the scientific community on COVID-19. Could you maybe just take us back and say, what's it like to be at the NIH when there's a new virus that's emerging and threatening the world?
Yeah, it was, it was first, we prepare for pandemics because we've had a few new diseases or new outbreaks that evolved over the past several years. There was Zika, there was acute flaccid myelitis in the United States over the past couple of years. Those were epidemics that emerged, and we always think about forming special teams and focusing on basic biology, supporting, better diagnostics, better countermeasures, and thinking about vaccines and other things that would prevent disease. So we were sort of used to it, but the scale of this particular one, this is the pandemic of a lifetime, and we had other coronaviruses and some coronavirus research ongoing, basic research in animal models and some possible therapeutics, but everything spread so quickly and threatened the United States so quickly that, it was very difficult to build up all of the research programs that needed to happen fast enough to make things happen and to develop therapeutics and preventative tools fast enough to have an impact.
So what were the first few things that you did? Did you look and see what research you already had ongoing that was relevant? Or, how do you get even oriented to something like this?
Yeah, well, we first looked at what had been done in SARS-1 and what was currently being developed for Middle Eastern Respiratory Syndrome virus, MERS, which is a coronavirus that isn't that closely related, but the idea was that some of the drugs and some of the animal models that things were being tested in might be very useful for this new coronavirus. So we first, started expanding upon the research base that was already built there, looking at things that antiviral drugs that were known to be effective or thought to be effective against MERS, and testing them as quickly as we could against SARS-CoV-2. We also very quickly, as soon as the genetic sequence became available, we tried to capitalize on some of these platform-based vaccine technologies, like mRNA was the first vaccine-type platform technology that was used to design a vaccine against the new coronavirus.
Can you explain a little bit what you mean by platform technology as opposed to other kinds of technologies?
Yeah, a platform technology in a vaccine is, a certain type of vector or a certain approach to making a vaccine that's the same across different pathogens and just uses, in the case of messenger RNA, you would just use a different sequence to make a vaccine against a different virus. And in the case of SARS coronavirus 2, as soon as the sequence was published and the spike protein sequence, which is the protein that's essential for virus entry into a cell, as soon as that was published, manufacturers who had developed mRNA technology for other types of pathogens used that sequence to make a vaccine that they thought could be effective against this virus. So it was using the platform that had been developed, the mRNA in a liquid or in a lipoprotein sort of particle, in order to design a vaccine that could be tested for this virus.
And how much of what you had to do is, call people up and say, hey, you have this platform technology, do you think you could use it? Versus people beating on your door saying, I've got this great idea, please give us funding for it.
Oh, I think it was the second approach was what mostly happened. I mean, scientists were on this really, really quickly. I think that, there had been investments made in approaches like the WHO had a plan developed for disease X, which basically this was that was the exercise that prepared people to some degree for dealing with this situation. So as soon as the virus was identified, a lot of scientists, particularly ones who had experience in working with coronaviruses, were already thinking about what they could do to make a vaccine and what they could do to identify drugs, either repurposing existing compounds or developing ones that were in some earlier stage of development, how they could develop them further for testing if they were efficacious against this particular virus. So all of those things were underway almost immediately, as soon as the virus became available.
And then the next thing that happened is that there were sick patients. And there are all these questions that are needed, to be explored, including what is this disease like? How does it affect people differently? What works and what doesn't to treat it? How did you have to move to set up clinical research?
Yeah, we knew right away that we wouldn't have time to go through the usual steps of, drug discovery, drug development, test them in animal models to see if they work against animals infected with this virus, because, very, very quickly people were, being hospitalized in parts of the world, and then very soon thereafter in the United States that didn't have any options for therapy. So we needed to identify compounds that had already been in human beings testing against other diseases and and if there was a sign that they would work against this virus and inhibit viral replication, and if they were known to be safe, then the clinical trials needed to launch right away to see if they worked in this particular disease. So that was the thought process that was applied, well, first in China, because there were some therapeutics like an HIV protease inhibitor. It was, lopinavir ritonavir that was being used in China based upon sort of that rationale. The drug remdesivir, which was the first large-scale clinical trial that my division invested in, had been used in people who were admitted to Ebola treatment units in Africa over the past several years. It wasn't proven to be effective there, but we knew that it was effective in vitro against coronaviruses, and we knew that it was safe. So the clinical trial that we launched, the efficacy study, remdesivir versus placebo, was based upon that rationale that it was safe in humans. There's supplies of it that are available, and we can test this in sick patients. We can start right now.
So let's talk about that study for a second, because that study obviously showed some beneficial results. Was that study pursued on the same normal time frame as the usual NIH clinical trial, or did it have to move super fast?
No, actually, it launched a whole lot more quickly than we probably have ever launched a therapeutic clinical trial before. There were actually, I think there was one case in the United States, a patient hospitalized with COVID that got remdesivir through compassionate use. It had been used, we knew that it was being used in China, that there was a clinical trial. So, and it was made available in the United States. But the company that makes it, Gilead Sciences, did want to do more clinical trials to demonstrate whether or not it had efficacy. So probably, I think the timeline was on the order of 2 weeks from concept to discussions with the FDA, submission to the FDA for an IND to do the efficacy trial and IRB approval. I think the race was, you'll recall that there was repatriation of the Diamond Princess cruise ship passengers who were sick and coming back to the United States, and the State Department was organizing this because they had to go to special hospitals that had containment units set up for them. And so there was actually a race to get the trial approved and a safe to proceed from the FDA so that those individuals that were sick could participate in the trial if they wanted to. So that was what happened probably in a little over 2 weeks it took to get that trial up and running to the point where the first patient was getting an infusion.
So you bet on a winner from the beginning, but not everything is going to be a positive trial. When you get started, you don't know.
That's right.
What other types of therapeutics is NIH studying right now?
Yeah, so the interesting thing about COVID-19 disease, which I don't think people had a full appreciation of just a few months ago, was, remdesivir is an antiviral, so it inhibits viral replication by inhibiting the RNA polymerase. But it seems as though many people, as they get sicker and sicker with COVID-19, with disease, have a lot of inflammation associated with the virus. So a lot of the things that happen that clinicians have observed, in people who are very ill, is that there's a lot of clotting and complications related to clotting events like strokes and MIs. There's a lot of lung inflammation. So there's this immune reaction that is, incompletely understood and not well described that seems to happen. The terminology that is used is like cytokine storm, which is what you think of oncology patients getting when they get, very, very, chemotherapy that their tumor is really sensitive to, and there's a lot of cell death, and the immune reaction's robust, and the cytokine storm is what makes them sick then. So that observation has been part of what clinicians have described in taking care of COVID-19 patients. So in addition to antivirals now, we're testing host-mediating factors that limit inflammation. So the other drugs that have sort of been repurposed for treating COVID-19 are drugs that are approved for use in rheumatoid arthritis patients, for example. One study that we're doing now uses baricitinib, which is it's a Janus kinase inhibitor that's used in rheumatoid arthritis. And that, in addition to remdesivir versus remdesivir alone, is the compared strategy that we're testing in a follow-on trial to the remdesivir trial. And other clinical trials, other companies are testing things like antibodies against IL-6. That's a mediator of inflammation that is measured to be very high in patients with COVID-19. And there are a long list of other host modulating factors that are being tested.
And then you have some antibody type therapies too.
Right. Yeah. So as soon as, when people are convalescing from COVID-19 disease, the companies and people that specialize in humoral immunity you can get plasma cells from people who are recovering and then use those B cells to identify antibodies that might have facilitated the recovery that might be particularly effective against, binding and neutralizing the virus. So that approach has been used in developing monoclonal antibodies that might now move into the treatment setting, or they might actually be used prophylactically to prevent people exposed to COVID-19 from actually becoming sick from the virus.
So at a minimum, we'll be getting a lot of results about clinical studies over the next few months. Would you say that's fair? And maybe within those results, we'll have more signs of progress?
I think so. There's a lot of tools that have been developed. There's a lot of, there are antibodies and there are other drugs and other types of compounds moving into testing. And yes, I think we can look forward to getting more answers over the next couple of months on how to treat this disease more effectively.
So in the time we have left, I want to ask you about prevention and vaccines, because if I'm not mistaken, the vaccine investments at NIH also are in your division. Is that right?
Well, they're definitely at our institute at NIAID, and the investments also are at the Department of Health and Human Services, the BARDA, the agency that's responsible for advanced product development. And together with NIH, we're making significant investments in vaccine development.
Got it. So you're part of the team.
Yes.
And so how does this vaccine development effort look different, from your vantage point compared to a traditional, vaccine effort, which might take 10 years?
Yeah, it's the pace at which things are moving is much faster than probably any previous vaccine development effort. Usually, you hear people say it takes 10 years to develop a vaccine, and that would be based upon a company or a developer making decisions sequentially. If the data looks good in small animals, then you move to larger animals. And if that data looks good, then you move into humans and you manufacture a small amount and then you move into humans. And if those data look good, you might go into phase 2 trials. And, at a later stage, you develop in the processes for commercial production because all those steps cost a lot of money. But now, with this, public health threat, the U.S. government has supported companies in developing all of those steps in parallel rather than sequentially. So, we're already planning our phase 2 and even our phase 3 trials while the data from the phase 1 trials were still coming in. And developers are investing in processes for commercial scale up of manufacturing even before we know that the vaccine truly protects or not. That's what it's risky from a corporate investment standpoint. I think we have to emphasize that we don't want to put participants in trials at any undue risk. So the standard safety monitoring and planning for that is always in place. But the investments that a company are made a lot sooner and a lot more aggressively than a company would ordinarily make if this were your usual vaccine.
And it's not just one, it's multiple companies all doing that at the same time.
You're right. It's a lot of vaccines. There's a lot of ideas for vaccines, and a lot of them are moving through the pipeline faster, we hope.
Do you think at some point there'll be a bit of a choosing that'll have to happen, that some of them are going to do better early on, and the NIH and others in this world that are trying to foster vaccine development are going to have to take a look at interim data and make a decision about where to really invest? Or do you think you can push a whole bunch of them all the way through to see what happens?
Well, I think we do need a lot of different vaccines. I mean, I think that everybody in the world probably, or at least every adult in the world probably needs this vaccine. So there will probably be some that are going to be better for older patients or older people, and there might be some that are better for younger patients. And there are some that will be more implementable in low resource settings. So I think we need a variety of vaccines. I'm sure that not all will make it to the finish line with highly effective protection and also high level of safety. So those will have to be weeded out, and it might occur in the middle of a trial that they get weeded out. I can't predict anything more in a more precise way than that.
Well, it is hard to predict. Some people can be enthusiastic about one treatment and then it turns out in the trial that it doesn't work. And then the reverse can happen. Sometimes people can be doubtful, but then the trial shows that there's a real effect. And the NIH is there supporting, high quality research so that we get the answers that we need to be able to advance clinical care and prevention. I guess what I would say is that where I have confidence is not in a particular drug, but in the fact that you're where you are and that we are able to apply the tools of science to this challenge. And so I really want to thank you for spending a few minutes with me and for all the work you and your colleagues are doing.
Thanks, Josh. It was my pleasure.
Thank you for listening to Public Health on Call, a new podcast from the Johns Hopkins Bloomberg School of Public Health. Please send questions to be covered in future podcasts to publichealthquestion@jhu.edu. This podcast is produced by Josh Sharfstein, Lindsay Smith-Rogers, and Lamare Morales. Audio production by Niall Owen McCusker and Spencer Greer, with support from Chip Hickey. Distribution by Nick Moran. Thank you for listening.